And we'll move on to the next speaker, which is Inbar Fischer from Tel Aviv University, and she will talk about hyperbaric oxygen therapy in autism from mouse model to human patients. Inbar, please.
Hello everyone, my name is Inbar Fischer. I'm a PG student in Dr. Boaz Barak's lab. Today I'm going to present a study in collaboration with Professor Shai Frati from Asaf Harofeh Hospital. This study focuses on hyperbaric oxygen therapy for an ASD mouse model. Our research mainly focuses on one of the high-risk genes for autism, the SHANK3 gene. The two main known causes for autism are environmental and genetics, and it's known that up to 25% of ASD cases have a genetic cause. Because SHANK3 is affected in 1% of ASD cases, it is considered to be one of the high-risk genes for autism. The SHANK3 is expressed in the postsynaptic density of excitatory neurons, and its main function is to bind the cytoskeleton to glutamate receptors, and therefore it has an important role in synaptic transmission, integrity, and in interaction between neurons. In our work, we studied the insertion G3680 mouse model. At first, a full SHANK3 gene deletion was performed, but in contrast to this model, the insertion G3680 has a human-based point mutation, and therefore it aims to capture more precisely the disorder, as there are human patients with this exact mutation. The mutation is the insertion of nucleotide G in a certain point in the gene, causing a frame shift and then a stop codon, causing a dramatic downregulation of SHANK3 expression. This mouse model was well characterized in biological, physiological, and behavioral levels, and in the study they saw and observed severe synaptic dysfunction by the observation of and measuring electrophysiological measurements, and in other biological aspects. And in here we can see an example that mice that are homozygous to the mutation have much fewer spines on their dendrites compared to wild-type mice. In the behavioral level, they found impaired social interactions and extensive repetitive behavior both exist in human ASD patients. Although this mouse model was well characterized, there is still no treatment for this model, for other models with SHANK3 mutations, and for human patients as well. The potential treatment that we are examining is the hyperbaric oxygen therapy, or HBOT, which is a type of therapy in which the subject is placed in a chamber with 100% oxygen gas and pressure levels of more than 1 atmosphere. It was extensively tested in different pathologies, in different animal models, and in human patients as well. And it showed that it can hasten tissue recovery and to improve different physiological aspects. Because the biggest advantage of this therapy is that it is a non-invasive treatment, it was also tested in different neurological conditions, in human patients as well, especially ones with an hypoxic state like a traumatic brain injury. In the molecular level, HBOT have shown to improve different biological pathways, and in here we can see examples that it can improve mitochondrial properties, to encourage angiogenesis, and to reduce neuroinflammation. We wanted to further investigate the mouse model that we study and to examine whether one of these biological pathways is impaired in the mouse model, so this treatment could be beneficial. In order to do that, we performed real-time PCR to see if there are alterations in the expression of different genes. As I showed you earlier, the mutation is causing a dramatic downregulation of SHANK3 expression, and in here we can see that it happens already in the transcript level, causing synaptic dysfunction. We observed also downregulation of the expression of two other genes that are highly related to hypoxia, One is HIP-1 alpha, which is related to oxygen deficiency, and the other one is VEGF-A, which has a crucial role in angiogenesis. Because of those indications of an hypoxic state in the mouse model that we studied, we decided to start the treatment. You can see here in the photos the system that we have in the lab. This is the HBOT chamber, and this is the placebo. The treatment will be in total 2 months, 40 sessions, 1 hour a day, 5 days a week. A month is already behind us. The HBOT groups are receiving 100% oxygen and pressure levels of 2 atmospheres, and the placebo are receiving air, which contains 21% oxygen, and pressure levels of 1 atmosphere. In our experimental design, we decided to test 4 groups, 2 groups with mice that are homozygous to the mutation, and 2 that are control littermates or wild type, and they are divided to either HBOT or placebo. We began the treatment when the mice were 1 month old, because this is a neurodevelopmental disorder and we want to start as early as we can, and we'll end the treatment when the mice are adult, are 3 months old, and already exhibit the hypoxic state that I showed you earlier. To conclude, we show indications of an hypoxic state in the insertion G3680 mouse model. Therefore, we decided to start HBOT and to treat this model with hyperbaric oxygen therapy, and to see its effects in the hypoxic state, and afterwards in the behavioral level. And hopefully in the future this study could be extended to human patients as well with different SHANK3 mutations and to other ASD-related disorders. So I want to thank the lab members, my supervisor, Dr. Boaz Barak, and for you for listening. So thank you very much.
Thank you very much, Inbar. I must know that you are the first speaker who made it in time.